In this video, we demonstrate a novel method of encapsulating protein drugs in polymeric microneedles. The fabrication of microneedles is divided into two stages: the fabrication of microneedle backing layer, followed by fabrication of microneedle shafts. Up first, we will describe the fabrication of microneedle backing layer. The pre-polymer solution containing protein drug bovine serum albumin is first wicked into the cavity formed by supporting cover slips on a glass slide substrate. This setup is then placed on a flat stage below a high-intensity UV lamp at a distance of 9.5 cm from the UV light source. It is then exposed to high-power UV light for 2 seconds to polymerize the liquid and form the microneedle backing layer. The glass substrate is then removed from the setup and the backing layer containing cover slips removed from the substrate with the help of a blade. This is followed by fabrication of the microneedle shafts. The pre-polymer solution is similarly filled in a cavity formed between the cover slip holding the backing layer and the glass substrate. The length of the microneedle can be controlled by the number of cover slip spacers placed on the base glass slide. One major difference between the fabrication of microneedle backing layer and shafts is the use of specially patterned photomask in this step. The photomask consists of a plus film which is inked in a specific pattern, leaving transparent dots in between for UV light to pass through. This film is placed on the cover slip on which backing layer is attached, and the setup is placed on a platform at a distance of 3.5 cm from the UV light source. It is then exposed to high-intensity UV light for 3.5 seconds to form the microneedles. The setup is removed from the stage and the photomask is removed with the help of tweezers. The leftover unpolymerized prepolymer solution is washed under running water and the microneedles removed from the substrate and let to air dry. The fabricated microneedles were tested for stability of encapsulated protein drugs by releasing the protein in water and testing its primary, secondary, and tertiary structural characteristics by SDS PAGE, circular dichroism, and fluorescence spectroscopy, respectively. The results show that structural characteristics of the protein were similar to freshly prepared solutions and significantly different from BSA degraded at high temperature and under acidic conditions. This in vitro release demonstrated that BSA was almost completely released at the end of 6 hours. In vitro permeation experiments using rat skin showed significant enhancement in microneedle-mediated protein permeations as compared to passive diffusion. Following this, biocompatibility of microneedles was assessed by mitochondrial dehydrogenase and lactate dehydrogenase activity using MTT and LDH assays, respectively. High cell viabilities were observed, indicating the biocompatible nature of polymeric microneedles.